Hello, everyone. Thank you for joining us today for our webinar. As you probably know, May is NF2 Awareness Month, and we are so grateful to our panel of expert speakers today who will be updating us on their work and the adeno-associated virus approach for gene therapy. We're also very grateful to our audience. So thank you all for taking some time to learn from us today and to support NF2 Biosolutions. I'm Mary Sell. I'm the parent of an NF2 warrior, and I'm a board member for NF2 Biosolutions. I have a few reminders for us before the speakers get started. This webinar is being recorded, and the recording will be posted on the NF2 Biosolutions website under the webinar section. You can submit your questions today during the webinar in the Q&A area of your control panel. So we'll be watching those questions as we go, and we'll be addressing most of the questions in the second half of the presentation today. We also have questions that we received when you registered. Captions are available, so on the Zoom Room control panel, you'll want to click on the captions icon. And you may need to click on the more icon first in order to see the captions icon. I also wanted to tell you about another project that NF2 Biosolutions is currently raising funds for, the Antibodies Project at Yale University. Researchers are developing an antibody to penetrate NF2 cells and promote tumor immunity with this project. And all of your donations, large or small, have a great impact and are put towards research for a cure for NF2. We're an organization of volunteers, so all of the dollars that are donated go towards NF2 research. And you can see on this slide what some of your dollars can purchase. For small amounts, like any amount, $5, $10, $50, those small amounts help cover lab materials like reagents or cell cultures. $200 can cover the cost of housing and evaluating lab animals for this antibody project, or $5,000 can cover a significant portion of antibody production and characterization. So again, your donations are so important, and we greatly appreciate your help there. I'm going to go ahead and introduce our panelists now. We have Dr. Catherine Meyer with us, who is the Chief Scientific Officer at Alcyone Therapeutics, where she leads the development of gene therapies and advanced delivery techniques for neurological disorders. With a background in RNA biology, she has over a decade of experience in gene therapy, including key contributions to the AAV9-based treatments for neurodegenerative diseases at Nationwide Children's Hospital and The Ohio State University. We're also very grateful to have Dr. Gary Brenner with us today, who currently sees patients at the Massachusetts General Hospital Pain Management Center, where he's the director of the Mass General Pain Medicine Fellowship. He's an associate professor in anesthesia at Harvard Medical School. Dr. Brenner runs a National Institute of Health funded lab focused on delivering gene therapy or developing gene therapy for the often painful peripheral nervous system tumors associated with neurofibromatosis. We're also very lucky to have Crizelle Alcantara with us today. Crizelle is a molecular biologist and NF2 patient dedicated to advancing research and advocacy for neurofibromatosis type 2. She leads translational NF2 research in the Meyer Lab at Nationwide Children's Hospital, focusing on gene and cell therapies. Crizelle also serves as a volunteer ambassador and a member of the Scientific Advisory Board for NF2 Biosolutions and as an NF advocate with the NF Network. So we will let our speakers take it away here.
All right, thank you, Mary. Crizelle, are you going to share the presentation?
Okay, now we're seeing it, your agenda.
Okay. I have some black boxes here on the screen. I don't know if you have two. Is it just on my screen or?
I'm seeing the black boxes.
Okay. Is this in presenter mode? Maybe let's try again. Sorry, I'm very excited to be here. So I'm going to give you a quick introduction on gene therapy, just to explain the concept in more detail. And then Chriselle, who did most of the work in my lab, a lot of work actually, will present you the data that I think is pretty exciting. Still the same. Hold on. Chriselle, do you want me to try? Share on my end?
Sure.
Okay, hold on. Okay. All right, how does that look? Better?
Yep, we're not
Okay.
seeing any black boxes.
Okay, good. All right, as I mentioned, I'm going to give you a quick introduction, but then I don't want to be in the way of Chriselle and Gary, who are going to talk about the data some more. So gene therapy, it's a very broad concept. It really means to use any technology needed or possible to deliver therapeutic genes to cells or tissues. Genes are the blueprints of the human bodies. They instruct the body and the cells, of course, what type of proteins or other small RNAs to make. And as you all know, these proteins are often very critical for the function of the cells. And there are many diseases, some of them are common, some of them are less common, that are caused by either faulty expression of proteins. Proteins are made somewhere where they shouldn't be made, or not enough of a protein is made, or a protein is made that isn't complete or has the wrong folding because it carries mutations. So all of this can cause disease. So there are different types of gene therapies. Gene replacement is a term that I think is actually not very good because it can be confusing. It just really means adding a healthy copy of the gene. We're not replacing anything. It's like putting a second car into your garage if your first car isn't working. Oftentimes nowadays, they call this also gene addition, which I think is the better term for it. There's gene silencing, where we have technologies where we can shut down the production of a toxic gene or RNA. And there is gene correction, which you might have heard about, which would really require to go into your cells and change the DNA in order to have an effect. So those are the differences between the different approaches. In many cases, we use viruses because viruses are very efficient in delivering genetic materials to cells. That's what they have evolved to do. And it can be a little bit confusing with the terminology. So real quick, a virus is usually what we all get, which is a wild type virus. For example, influenza, when you get the flu, that's a virus. But when we use viruses, it's really just a delivery box. We remove all the genes that make out the virus, and we put in the therapeutic genes under a control element so that whatever is delivered to the cells has nothing to do with the original virus. We just use it as a package. And when we talk about these viruses, we often call them vectors or recombinant viruses. So that means just we have modified them to deliver a payload that we want to express in the cells. Adeno-associated viruses that have been discovered in the 60s really have changed our capabilities, especially for targeting of large organs such as the nervous system. There are some important differences in AAVs compared to other viruses that make them ideal for delivery of genes. Number one, they are not known to cause any disease in humans. So we don't have to be worried about diseases that could be carried in these viruses. And number two, the modified versions that we use, they don't replicate. So once we infect the cell in your body, the message stays inside, but it doesn't infect other cells of the body. So we don't have to be worried that it spreads to a place where we don't want it to be, other than, the initial load. So AAVs have really become a to-go-to technology for many gene therapies that happen inside the body where you can't take out the organ, but you have to address things locally. And there are several AAV gene therapies that are now approved, both in US and EU, for example, treatment of SMA or for treatment of retinal disorders, etc. I don't think I have to spend too much time, unfortunately, talking about this. You all know that the current options for NF2 patients are not sufficient, and they have severe side effects on their own. They can also not prevent future tumor growth. It's really more to kind of try and address the symptoms as they come along. So we were really interested in seeing if there are safer, more effective ways to treat NF2. And so what we developed is a gene therapy in which we express the human NF2 coding sequence under a truncated version, a shorter version of the NF2 promoter. We really wanted to use the promoter of NF2 because we think it's important to express this gene in all areas and in all cells that would naturally express NF2. What's important also with this construct is that every aspect of the construct except what is specific for NF2 is already in clinic, has already been used in hundreds of patients, and is being really safe and well tolerated. And then again, just to kind of point out, this is called gene replacement, but we are not replacing anything and we are not interfering with the endogenous genes. We are simply bringing in an additional vector, an additional copy of a gene that will make the healthy NF2 protein. So I'm going to hand it over to Chriselle from here. Chriselle, just let me know when you want me to switch the slides.
Okay. Thank you so much, Kat, for that wonderful introduction. Hopefully everyone is on the same page now on what we're trying to do here with the NF2 gene therapy. So what we first wanted to test was after we produced the vector, we designed to express either the green fluorescent protein as a reporter or the NF2 protein itself. We injected them into mice to answer the question of number one, is the vector actually delivering the transgene into the relevant tissues and subtypes where we want the transgene to be expressed? And number two, more importantly, we want to see if the vector actually reduces or shrinks the tumors in mouse models of NF2. Next slide, please. So first we did injections into wild-type mice. When you say wild-type, these are just normal mice without any genetic defect in them. With the goal of just seeing if we are targeting expression of the virus-reduced genes into the tissues that we want to be targeted by the promoters expressing the transgenes. So because we are using the NF2 promoter itself, we have data where we expect the NF2 promoter to be expressing all throughout the body of the mouse. So in the mouse brain, which is the relevant tissue that we look at to study NF2, we are seeing a lot of GFP or green fluorescent protein receptor expression in the brain regions where we do expect NF2 to be highly expressed in the brain of the mice. So we also did co-expression studies with another protein called S100 beta, which is actually a marker for Schwann cells, which is the most relevant type of cells in NF2 from which the tumors actually arise in this disorder. And if you are seeing yellow signals, that means the green overlaps with the red, meaning we are actually targeting the correct cell types, not only in the correct cell types, but also the regions of the brain where we want the transgene to be expressed. Next slide. The same goes with the vector that is actually expressing the NF2 protein, the transgene NF2 protein. we are also expressing it in the relevant brain regions as shown here, and also supported by other assays that shows the protein and messenger RNA levels. Good, please. Just to confirm that we are, yes, expressing the transgene in these brain regions. Next slide, please. Now we know that NF2 tumors also arise in the meninges in NF2 patients, resulting in two meningiomas. So we also want to see if the vector is also able to express the transgenes in the meninges of wild-type mice. And again, here, if you can see yellow, where there is an overlap of NF2 transgene expression in the markers for the meninges, that means we are indeed targeting the relevant cell types in the meninges of the wild-type mice. And lastly, we also, because tumors also form in the peripheral nerves of the NF2 patients, we also wanted to see if the vector is able to target the peripheral nerves in the wild-type mice. And similar to the previous two slides, we are seeing a lot of yellow signal here, where the GFP reporter gene targets the NF2 protein in the cystic nerves of mice. So we also injected the vector in a mouse model wherein tumor cells were injected under the skin of mice and let them grow into tumors, and then injected them with the virus. We monitored the growth of those tumors compared to a vector, an empty control, sorry. And as you can see here with the graph, there is significant reduction in the growth of the tumors in the mice that were injected with the NF2 expressing gene therapy vector. We also wanted to test not only in the mice, but also in relevant cell types or cells in culture, which included patient cell lines that are derived from actual NF2 patients, also human primary tumor cells from vestibular schwannomas, and lastly, a cancer cell line that has reduced expression of NF2. And the following slides just show here that indeed these cell types have reduced expression of the NF2 protein. Yeah, as shown through imaging and other assays. And that treatment of these cells with our NF2 gene therapy vector actually results to phenotypes that rescue these cells from being tumor-like to a more healthy physiology. So this would be results that we got from NO2 patient-derived tumor cells. And then the next slide would be similar treatment that we did to human vestibular schwannoma tumor cells with reduced NF2 expression. And again, treatment with the vector results to healthier readouts from these cells. And lastly, we also tested our vector in a cancer tumor cell line that has reduced expression of NF2, just to show that this therapy can actually be expanded to other diseases as well, including cancers where NF2 expression is aberrant or abnormal. And this just shows that the cancer cell line actually has reduced proliferation levels after treatment with the NF2 vector or gene therapy that we have designed. And next slide, please. Okay, so I think we can, I'm not sure if we can conclude already here or if Dr. Brenner would like to share his results first with our NF2 vector.
You're muted, Dr. Brenner.
I just have a couple slides. It's a pleasure to be here. And I'm just going to briefly present some confirmatory in vivo data using a human xenograft model of NF2 related schwannomatosis, or NF2 disease, as a lot of people know it. And basically, what we do is we take a human-derived NF2 schwannoma cell line and implant it in the sciatic nerve of mice. You have to use immune deficient mice or they'll just kill the human cells. That's why you see what's this hairless mouse called a nude mouse. And when you put the tumor cells in the nerve, which is what we think of to be a biologically relevant environment, you don't have to add any growth factors or anything as you might have to do for subcutaneous tumors. They just grow fine. And they have a reporter in them, so we can make them light up and use a very sensitive camera and follow the tumor growth over time. And it doesn't grow exactly like a human tumor. The tumor cells fill up the sciatic nerve. You can kind of appreciate this is a normal sciatic nerve, and this is a sciatic nerve full of tumor cells, but we can use this light signal to follow growth over time. So we implant, we let the tumor develop, we inject either the vector or control substance, just basically salt solution, and then continue to follow. And these are the kind of results we get. And it is highly significant. The black line is how the tumor grows if you just inject the vector into the tumor, the PBS into the tumor. So that's just normal growth. And it's over weeks. And this blue line is how the tumor grows if you inject this NF2 construct for addition, like Dr. Meyer. I like the term addition since we're not actually replacing anything. That's a totally different technology. And there's a quite clear result. And we had a second control in this experiment. You'll see the blue line's the same. And the second control is AAV vector that instead of delivering Merlin or NF2, delivers green fluorescent protein. And it's important to have both these controls because actually this protein can be toxic to cells all on its own. And what you see is, again, there's a significant difference between the control, which is the green protein, and that same growth curve for the NF2 addition. So there's kind of two controls, which is always good. And that's all I have. We're going to look forward to further replicating this with vector and hopefully get to clinical trials naturally.
Very good. Thank you all for presenting where you're at with your research and all the hope you're giving us all for curing NF2. The next part of our presentation today is going to be a panel discussion. So I'll just be asking some questions, and I'm not sure who each question is best targeted for, so you guys can decide or each answer. And we'll also throw in questions from the audience. The first question, what makes AAV9 a promising vector for targeting NF2 tumors specifically?
Maybe I can start, and then Gary, you can maybe chime in. So AAV9 is a vector that has been used in the clinic for a long time. There is over 3,000 patients at this point that have been treated with AAV9, and generally it's been well tolerated and safe. AAV9 is also very good at penetrating tissues and getting to the places where it needs to go. So those are the things I really like about it. So this gives us the opportunity to not only treat the tumors, but also think about NF2 as a genetic disease and maybe think of a more systemic prevention study where we could hopefully reduce number of tumors that might develop in the future.
Yeah, naturally, I agree with everything you said, as I always do, and presumably always will. I would just add that, as Kat indicated, there's reason to think that AAV9 could be a good capsid vector for use with systemic therapy. And while our initial enabling data is with direct intratumoral injection, as everyone knows, almost all of the individuals with this disease have multiple tumors. And certainly some of the tumors, for example, if they're near the brain stem or in the brachial plexus, which is a mass of nerves kind of near the armpit, surgical resection either isn't possible because it's too dangerous, or it's just so highly associated with potential neurologic deficits that people really try to avoid resection. So while I think it's important to have enabling data with direct injection, from a treatment standpoint, if the gene therapy can be given systemically, that's going to be much more optimal. And certainly, as Kat alluded to, absolutely necessary if it's ever going to be developed as a preventive therapeutic. Now I've just repeated everything you said. Sorry, pretty much.
Yeah, and I think maybe this is a good moment to add. There's also one question in the chat about AAV9 toxicities. So that might be a good moment to just do a quick comment on there. So I can't speak to why CTF has changed their funding strategy, but as I said, AAV9 has been used in thousands of patients very safely. However, there are, certain barriers and certain toxicities that have been observed, in some rare cases fatal, which have to do with really high doses that were delivered intravenously in most cases, or really, really high doses that were intrathecally delivered in one patient. We do know the boundaries better and better with every day for AAV9, and I think we can, have effective treatments based on the data that's coming out of multiple clinical trials. Taysha just yesterday announced positive clinical trial data for their RED program with AAV9. So yes, anything taken at too high levels can be toxic and can cause severe damage. So this is something that is known, but we do understand it better and better with every day, which is another good part about AAV9. There's a lot known about the pathways and the doses that are safe and well tolerated.
Okay, thank you to Tawny for sending in that question about asking why CTF has withdrawn funding for AAV9 treatment. Tawny sent in another question for Dr. Brenner. She says, I think you presented about 2 years ago on a vector treatment and the mice were displaying shrinkage of established tumors. Is this the same treatment? This appears to slow tumor growth rather than shrinking tumors.
Sure. So our lab has developed some very different strategies in which we deliver pro-inflammatory proteins or genes that become proteins. And the idea was we wanted to develop a host, or the animal develop an immune response against the tumor. What we actually found was these, what are called inflammasome components, are very efficient at causing programmed cell death, but at least in mice, not a immunogenic or pro-inflammatory type of death. So, providing a functional copy of Merlin or NF2, which is what we're talking about today, is entirely different from delivering overexpressing an endogenous protein, which is what we're doing with delivery of it's called ASC or gasdermin. They're both parts of the inflammasome. I'm very philosophical about animal efficacy data. Mice aren't humans. Inbred mouse immune systems aren't even the same as the mice that are scurrying around in your house during the cold season. So I think it's very important to see that in animal models, there's an indication that these treatments could be effective and develop the science. But at the end of the day, you have to test these products in human beings because how they act in humans, the biology in humans very well can be different. It can be less effective. It can be more effective. Like what we're doing with those inflammasome products in a human, maybe it will be immunogenic. Who knows? And that can be good and that can be bad. So what we do is, and Kat, you can say just the opposite. It's okay with me. I think the most important thing is to have biology that makes sense based on both what you see in human tissues when you can get them and what you see in animal data, and do your best to de-risk adverse effects. And as Kat said, one of the best ways to de-risk is clinical experience, which AAVs are very, very safe. It's tragic when a treatment kills a patient. Tragic, obviously. Almost every treatment we have has the potential to lead to, I'll just say it, to lead to death. We have many drugs have very, very rare adverse effects that can be fatal. But you have to look at the likelihood of benefit and the likelihood of harm. Crizelle, Kat.
Yeah, I think it's very well said. I don't think I have to add anything. And I think maybe one thing to say is that these different therapeutic strategies could be combined in the future as well, right?
That's what we always think. It's like I'm almost afraid to say that, but it's entirely sensible. And there are delivery technologies that could facilitate that. Yeah. There's barriers to combining therapeutic strategies, but it could make sense for sure.
Right.
Yeah. Oh, go ahead.
Yeah, I was just going to ask you, Mary, if you want to continue with the questions or if you want to, how the plan is to moderate this.
However the flow goes best. We're learning from you guys. I could throw in another question to kind of tack on what we were talking about. What challenges have you faced, or what do we expect to face in transitioning your research from the lab to a clinical setting? I know you talked a little bit about that, but what other challenges are we expecting?
Yeah, so from where I stand, and that's my bread and butter, translating programs from the preclinical stage into the clinical stage, and then maybe Gary, you can talk a bit from the clinician side. I really don't think there are big barriers. The main issue is always, funding, because the production of clinical material, clinical vector, is expensive. But we've already had a pre-IND interaction with the FDA. I think our plan looks very good to the FDA. Again, if we're starting with intratumoral injections, the doses compared to what we would use for a systemic, for example, spinal fluid delivered program, are so much lower that we don't really anticipate big hurdles, which goes to one of the questions also in the audience. We think, based on the data that at least we have from Gary's studies and the studies at Ohio State, that the amount of virus that we need to see an effect is very feasible. And we have also performed some safety studies in healthy mice, and we haven't seen any toxicity or off-target effects so far. So I think this is actually relatively straightforward from a translational point of view in terms of experiment. What is always the big thing is the funding and being able to move this forward and get, a clinical site set up, etc. Gary, did you want to ask anything?
The
To add
first,
anything?
yeah, the first three thoughts that came to my head were money, money, and money. And it's really sad. The current situation for early phase biotech companies funding is not good. It hasn't been good for a while. We all know what's going on with our executive branch and funding for basic research, which can facilitate translation to clinical trials by de-risking the program and making investors more likely to risk their monies. Other than that, I agree with Kat. I think that the data that we already have is very strong in conjunction with the history of use of similar products, at least the vectors.
Is there any way to identify a timeline from moving from preclinical trial to clinical trial, or is it all dependent on funding? Oop, Catherine, you're muted.
Sorry, here I'm thinking and the timeline is strongly impacted by the funding that's available. And, so it is possible to move a program into clinic from the stage we are at within a year and a half, if the money is there. And the problem is sometimes at certain time points during the project, small amount of money, which is very important for many projects, but so at some key direction points you need a bigger chunk of money. Like if you want to reserve for clinical manufacturing, you have to put down a certain amount of upfront money, else you cannot do it. But what is good for us is that there is a lot of competition when it comes to manufacturing in the AAV space right now. And that has been helping to bring down the pricing significantly. It also forces the manufacturers to offer more flexible payment plans and have other, flexible strategies that make it more attractive in terms of attracting new clients. So that's good for us. And hopefully we're going to see more of that as well.
Okay, and you've already touched on this a little bit, but I'll keep going through the questions. And how does the NF2 gene addition strategy differ from gene editing or other gene therapies being explored? How is gene addition different?
Chriselle, do you want to take this one, or do you want me to go ahead? I can do it if you. Yeah. So and that's exactly why I don't like the gene replacement term. So gene editing is really when you go in with an enzyme, a protein that will take your DNA, your genome, will break it and will change it, will remove the mutation, or you can add in a gene into your DNA. The gene addition does not integrate in your genome. It doesn't touch anything that belongs to the patient's genome. It just basically adds almost like an additional chromosome, a piece of DNA that will produce this protein in the cells. And therefore, the hurdles on getting this into the clinic are a little bit lower because the risks of damaging other genes are a lot lower compared to gene editing.
Yes.
Okay.
I have another comment on that, if I may. Because I work with CRISPR gene editing technologies now too. And we have done some initial studies on NF2 patient cells. We found out that it's actually not efficient to use CRISPR on these cells because their own machinery is defective, which is important when you are doing gene editing using CRISPR, using the cells machinery. We found out that the NF2 cells are actually defective, and that's why it's not like working very efficiently in these cells, which is very interesting.
That's a good point also.
Okay, another question from the audience. When the therapy becomes available, the gene therapy, what would be the cost per patient theoretically? What ages of patients would be able to participate in a trial?
Yeah, I think that really depends on the approach and the interaction with the FDA as well. Sometimes, of course, it depends on the clinical trial design as well, but in some cases, especially if you go for accelerated approval, you have some limitations. They might say, yeah, we're going to give you approval for just a certain population of patients at this point, while you continue to prove your drug in a broader patient population. So I think it's too early to say where this would be going. For that we would have, I think the first step is to gather initial clinical data to be able to continue these discussions and move forward from there. Gary, did you want to add anything there?
Not a whole lot other than, in my view, the first patients to be treated would be adults to assess any toxicity. Ultimately, any of these, I don't see any reason why there would be a likely age restriction if this were effective on who could be treated.
Yeah, that the FDA in many cases recommends or highly recommends adult patients if they are available in order to establish informed consent. With children, pediatric populations, it's a little bit more difficult. If they cannot consent on their own, the parents are responsible for them. So that makes it more complicated.
Another question from the audience. How long does gene therapy last? Does it repair our genes and remove this mutation? Is it permanent?
So this type of gene therapy is not integrating into the genome in most cases. To some extent, it potentially will. We don't know how that will be happening in the tumor cells. We would need more detailed analysis. We don't know exactly how long it will be lasting. In dogs and other large animals, we know past 10 years that the expression hasn't changed. So it's certainly very long lasting. Now, tumor cells that divide might over time lose the gene therapy, but the good part is in this case, you can actually redose because it's not an issue for you if there is some immune reaction to the tumor cells, right? So that could actually be a good thing and might even improve the treatment as well. So we can't really say how long it will last other than based on what we know from the clinic, it does last a long time. It might not be forever. We might have to redose, but it could be several years.
Yeah, just add a couple things. So when the tumor cells divide, they kind of share whatever of these gene therapy products are in them as they divide. The animal models, the cells divide much faster than the human tumors because they're almost cancer-like in the rate, how fast they divide. They don't metastasize or invade local tissues. So my point there is the fact that we see any efficacy signal in these very quickly dividing animal models, I think is a good sign that in human tumors, where the cells divide less, so they're going to hold on most likely to the product longer, there's more likely to see a positive effect.
Okay, thank you. I'm going to combine a couple of questions. Monica in the Q&A panel asks, is it possible to use this kind of gene replacement when not one altered gene of NF2 is found? And another person asked, could this therapy be used for mosaic mutations?
So yes, because it is not specific to a certain mutation. I think, we could use it for any disease that is caused by a reduced level of NF2. It doesn't even necessarily have to be Merlin related. Technically, any cancer that will benefit from reducing cell division, if we overexpress Merlin, we would just have to measure the response, of course. But yes, so technically we don't necessarily need a specific mutation or specific case. We could use sporadic schwannomas could benefit from this as well. Yeah.
Okay, David asks, have you thought about screening for NF2 enhancers? This could boost the expression level of vector-driven NF2 expression and improve efficacy. Any comment on that?
So we have not. We're using a form of the endogenous promoter, as said, because we are trying to mimic the expression that is found in the body. Certainly, this is something that could be done in the future, but again, necessarily sometimes massive overexpression is not always a good thing. So we'd have to be careful adding such reagents to the mix. Yeah.
I want to loop back to a comment Kat made just a few minutes ago because I realized there's two things. One is redosing naturally could be very important because you always hope you totally eliminate tumors, but I'm not sure that that's, while hoped for, entirely rational with this approach. So redosing is important for that reason, and also people develop new tumors, as you know. And beyond that, there's AAVs in the universe around us. So some people show up already, they've been infected and they have immunity. We did a study where with our other gene therapy, which I think is likely applicable here because it was the same intratumoral injection. And if you make an animal immune to the AAV vector, it has zero effect whatsoever on the efficacy. My thinking is that while we can show there's antibodies that can kind of attach to the AAV and neutralize it, you inject enough into the tumor environment that maybe some of it gets neutralized, but at the end of the day, you just overwhelm that. I do wonder if you were going to give the virus systemically, like intravenously, that is, I think, more likely to be an issue. But people, Kat knows this better than I, are developing all sorts of technologies to make these capsids, these vectors, minimally immunogenic or not really seen by the immune system. And so there certainly could be workarounds. And also, we don't know if we give it through the cerebrospinal fluid, how immunity might affect things.
Okay, we have a few more questions about the toxicity, and I know you guys have touched on that. Alex is asking, have you checked the selectivity of your NF2 PROM-A prompter in common AAV9 off-target organs?
So as I mentioned, we did safety studies already in mice. We haven't seen any toxicity in any of the organs that we looked at. So we're pretty comfortable with the construct that we have. Of course, the next step will be a large animal safety study, which is one of the items, right? That is part of the translational studies as well.
And I would say we didn't see any what you call behavioral toxicity in the mice. And remember, for the nervous system, it's kind of like a worst case scenario. We're injecting this vector directly into the nerve. That's not something you'd expect with human beings where typically as the tumor grows, because it's from not nerve cells, but supporting cells, the Schwann cells, generally it kind of pushes the nerves more or less out of the way. So if you inject directly into one of these, into a schwannoma in a human, you're I would think generally not likely to inject directly into a nerve. And with image guidance, it's probably quite likely that you would avoid injection into a major nerve. Like we all worry about the cranial nerves or big, major nerves like the sciatic nerve. So I think that would be avoided. So I think that's a very positive finding.
Yeah, and even with the injection into the spinal fluid that we did with our vectors, we didn't see with that, which is basically systemic delivery. We didn't see any issues. So I think we have a construct that is very well tolerated, even in wild type mice, which have the normal levels of NF2 already.
Okay, and Mary also tacks on to that discussion. It was mentioned on the CCF webinar, and it has been reported in the news that many companies have stopped using AAV due to toxicities and that the vectors are not reaching enough cells. Do you have any comments on that? Any other comments?
Yeah, I mean, I think you have to put this also a little bit in the context of after the approval of Zolgensma Luxturna, there was a huge excitement. I don't want to say it in a negative way. You could maybe say hype. People were very excited about AAV, and a lot of investments were made, and a lot of companies were formed based on very little data, right? And so after a certain amount of time, it was probably to be expected that some of these things, these amazing things that people invested in are not going to work out, right? So what we're seeing now, I would say, the two things that we see is that the bigger pharma companies are being a lot more careful getting involved. They want the smaller pharma that is more specialized to really figure this out before they get involved. And then a lot of small rare disease programs have unfortunately been returned because there is some, I think, issues that the field has to work through in terms of reimbursement strategies and how you price the therapy, but also how do you run a clinical trial in a very small disease area where you have maybe 500 patients and you have to treat 100 patients in your clinical trial in order to get approval, and then weighing that against manufacturing costs, right? So those are the big problems that we see. However, I would say in this year and last year, end of last year, I see significant upside. I see more excitement. There are some exciting data coming out from clinical studies from different groups. As I said just yesterday, announcement from Taysha on Rett syndrome looks really, really exciting, I would say. That we're working through these things. And the FDA is also showing more openness to smaller clinical trials that don't require, for example, necessarily a placebo control, which is something that we've been struggling with. So I think we're going to see a good amount of strong AAV programs make it through. But I think we're just in this time where things are just separating from the maybe a little bit overinvestment, if you can say so. Yeah.
Okay, and we have just a few minutes left. I want to go through a couple more questions and then give you each an opportunity if you have any closing comments. David asked, are you planning to test this candidate construct in a transgenic NF2 knockout mouse? I believe Marco Giovanni has developed a model.
So I'll start with this. There are a variety of genetic models for NF2 related schwannomatosis that involve knockout or an activation of the NF2 gene. They're hard to use, and it's hard to predict when tumors develop, exactly where they'll develop. Very expensive models for looking at certainly direct tumor injection because you really have to do MRI to see where they are, and that's expensive because you have to do generally MRIs over time. And then some of the tumors may not be in locations that you can really feasibly inject. And I think it's a very reasonable question. And it's certainly one I've gotten with grant reviews plenty of times. But I would go back to all the models, animal models we use are approximations and don't tell us how these products will behave in humans because the biology is different. Could you make a claim that the biology of these genetic mouse models is closer to human than implantation models? Yeah, maybe. But I still don't, none of this gets you to the answer, is this something that's going to help patients? And beyond that, you do want to test in patients because sometimes you learn from that. And if you have the funding, there's the potential to make a better second generation product. And I don't think you're ever going to get there with these mouse models. I don't know, Kat, maybe you have a different view.
Yeah, no, I think you're saying it very well. I think the next key experiment is testing this in larger animals to make sure the construct works as expected in a large animal, which is more similar to humans. We've done patient cell lines, we've done human tumor cells, we've done 2 different mouse models. I think the information that we could gain from an additional mouse model is very limited at this point, you know.
Yeah.
So I'm more excited about large animals and hopefully being able to try this in the clinic as soon as possible.
I agree. And if you were going to test this in a quasi-genetic model, no offense to my dog who's sleeping behind me, dogs develop nerve sheath tumors and they're spontaneous. Certain breeds develop them at a higher frequency. I think that potentially would have more value than a genetic mouse model. Though I still think there's enough information already there, and what you do is you de-risk toxicity to the best of your ability and try to get the first in human trials.
Very good. I want to give you all a chance if you have any closing comments to make a last comment. I do also want to tell you from the bottom of my heart, as a parent of an NF2 warrior, I can't thank you all enough for what you do every day and the hope you give us. And I know we're going to get to that cure. So thank you for your work. Any final thoughts before we let our audience go?
I mean, I always really like to thank the families and the patients and, for their willingness to deal with us researchers and, donate not only money, but donate tissue, blood, anything for us to even run these studies. A lot of the information that we gain depends on the families and the patients. And then on top of that, we're asking you to be the guinea pig in the clinical trial. So I know it's a lot. But it is the only way that we, I think, can make significant advances in the field. And, everyone is participating with these donations, be it blood, skin, anything. And so thank you for that. I really appreciate it. I have a lot of respect for all of you.
All right. Well, thank you. Any other words?
Well said, Kat. Appreciate those thoughts. And I think just so you understand, as researchers, I think the grand majority of us devote ourselves to try to help people and decrease suffering. And that's really what we're in it for. And we, like Kat said, your efforts are really greatly appreciated because from a selfish perspective, you help us work to facilitating our personal goal of helping people.
Very good. Well, thank you all for your time today. Crizel? Yeah.
Yes. Well, just to echo what Dr. Brenner and Dr. Meyer have already said. Well, as both an NO2 patient and an NO2 scientist, I also really want to thank you all for all the support that we had to get this project going. I'm really grateful for the opportunity to be working on this project, and I'm excited to see where we are now and where we are going, because this is really my dream too. So thank you so much.
Okay, thank you, everybody. We'll close it down now. So enjoy the rest of your day, and thank you for that hope that you inspired us all with.